Hello students, in previous class we studied the pollination. Pollination that is of three types: autogamy, kinetogamy and xenogamy, and other topic that was that agents of pollination. The different types of agents of pollination is there, that is wind, water, insect, birds and other animals. Now today we discuss about the outbreeding devices. Outbreeding devices. What is the outbreeding devices? Outbreeding devices, the such type of devices which is prevent the self pollination and enhance the cross pollination. The devices that help in the cross pollination, that is known as outbreeding devices. So different types of outbreeding devices is there. Suppose flower is bisexual. Bisexual means both the sex organ, that is male part and female part, both are present in the same flower, that is bisexual. But in bisexual also cross pollination is there. It is of bisexual flower is of two types: homogamy and heterogamy. Homogamy means both male part and female part mature at the same time, that is the homogamy, and which is leads to self pollination. But the heterogamy means both the sex organ, that is male part stamen and female part, that is carpel, is not mature at the same time, that is of heterogamy. It is of two types: protogyny and protandry. Proto means first, gyne means gynoecium. Protogyny, that means gynoecium mature first. Till that time, androecium is not mature, stamen is not mature, pollen grain is not mature. So, which prevent the self-pollination. And protandry, prop means first and andry means here, that word is utilized for androecium. Androecium, that is the male reproductive organs. So here the androecium mature first. Till that time gynoecium is not matured. So that also prevent the self-pollination. So protogyny or protandry may be occur in the bisexual flower due to heterogamy. So which prevent the self-pollination. In bisexual flower, one more condition is there, that is chasmogamous. Chasmogamous means open flower. If the flower is open and that receive the pollen grain from the different flowers, it's not same flower. And another condition, that is of clistogamy. If clistogamy is there, clistogamous flower is there, means always self pollination, leads to a self pollination. So in bisexual flower, chasmogamous is there, which is also enhance the cross pollination. Another is of self incompatibility. Or we can say self sterility. Self incompatibility or self sterility, it is due to genetic reason. Due to genetic reasons, sometimes the self pollen grain reaches on the self stigma, and the pollen grain of the same flower reaches on the same flower stigma, but that will not germinate due to self incompatibility. Means these two gametes are not compatible, which is generated from the same flower. It is genetic reason. It is due to genetic reason. Due to genetic reason, the self pollination is not occur. It is also known as self sterility. Self sterility means sterile, means that particular portion is sterile, or pollen grain is of sterile, which not germinate, not produce pollen tubes. So if the pollen grain transfer from the same flower anther to same flower stigma, that will not germinate, that is known as self sterility or self incompatibility. Another is of unisexual flower. Unisexual flower. This condition is known as monoecious. Monoecious, when two different types of flower, that is either male flower or female flower, present on the same plant but on different position. Here it is the male flower is there, or here it is female flower is there. These are two present on the different position. That also increase the increase the cross pollination. That is the out type of outbreeding devices that prevent the self pollination, that is unisexuality, just like in maize, castor, cucurbits. These are having the monoecious. They having two different of flowers on different position on different branches. Another is of dioecious. Dioecious, that means unisexual plant. Unisexual plant. If a plant having only one type of flower, only one type of flower, that one is the male plants, another is of female plants. One is the male plants, another is of female plants. In such condition, self pollination is not occur. So just like in papaya, that unisexual flower is there, male papaya and female papaya is there. So in these plants, self pollination is not occur. This is the also device for the outbreeding device, or which is enhance the cross pollination or prevent the self pollination. That is also known as dioecious. This dioecious is also known as dioecious, and it's only one type of flower present on the one plants. So that plant is male plants and another plant is of female plants. So these are the different outbreeding devices. These devices prevent the self-pollination and increase cross-pollination. So self-pollination also induce the inbreeding. Inbreeding means due to self-pollination continuously for from many till the many generation, that decrease the vigorous condition, decrease the some extra characters, due to which genetic variability is also decreases, or we cannot produce uh, good type of characters, good characters plants or good characters fruits. So outbreeding devices also decrease the inbreeding depression. That means due to inbreeding, due to self pollination, many good character will be eliminated from generation to generation. So these are the outbreeding devices, that is chasmogamous flower, heterogamy, that is proteny and protogamy, self incompatibility, monoecious flower, that is unisexual flower, and dioecious, that is unisexual plants. The next topic is of pollen pistil interaction. Pollen pistil interaction, uh, after the pollination, the interaction between the pollen grain and stigma or female part, all these interactions include in the pollen pistil interaction. After the pollination, that does not guarantee that right pollen grain is transfer on the right female parts. It may be same flower, which is the self incompatibility, or it may be from the different species. So pollen pistil interaction will start if the compatible gametes will be transferred from anther to stigma. If the wrong type of pollen grain will be reaches on the stigma, that will not germinate and they will not produce the pollen tubes. After reaching on the stigma, pollen grain absorb nutrients and swell up. absorb nutrients and swell up. If the right pollen grain will be transferred on the stigma, that will be absorb nutrients and swell up, and after that different interaction will be there, and different types of chemical will formed inside the stigma and pollen grains. If the right chemical will form, that will be accept the pollen grain. If the right chemical will not form, that will be rejected, is the rejection of the pollen grains. So the pollen grain will not germinate. Means here this interaction is in positive manner, that pollen grain will be germinate to form pollen tubes. If this interaction is not positive, that that is recognition of the pollen grain is not there, right pollen grain is not there, that will be not germinate. Before pollination, that pollen grains divided, which is on the in two stage condition. One is of, this is of generative cell, and this is of vegetative cells. Sometimes pollen grain reaches uh, on the stigma in three cell condition. These two are the male gametes, and this is of vegetative cells. If the right pollen grain will be reaches on the stigma, right pollen grain will be reaches on the stigma, this pollen grain form pollen tubes through the germ pore. Pollen grain form pollen tube through the germ pore, and that vegetative nucleus come into the pollen tubes, that now is called tube nucleus, and behind that generative nucleus come down, and which form two male gamete. Male gamete. So this is the stigma, style, and ovary. If the right pollen grain will be transferred, it germinate to form pollen tubes towards the ovule. This is the ovules, and it reaches towards the micropyle end of the ovule, micropyle end, and that micropyle end, here this is the micropylar end, micropylar end. Here one is the egg cells and two synergid cell is there. Two synergid cell is there. These pollen tubes move towards the synergid cells, and synergid cells, that is also known as filiform apparatus. Initially, this tube nucleus guide the pollen tube towards the female nucleus or egg cells, and it move inside the ovules through degenerating synergids or through degenerating filiform apparatus. And from this stage, means from this stage, germination on pollen tube until this stage, the all the events which is occur on the stigma, that is occur in between the pollen pistil interaction. All the events is occur in the pollen pistil interaction, and it is require different times. Means the pollen germination require different times. In some plants it is immediate require within 5 minutes. In some plant it is required 8 to 10 hours. Just like in chickpea plants, pea plants, crotalaria, balsam, it is germinate within the 15 to 20 minutes. We can also germinate the pollen tubes inside the laboratory in these plants, crotalaria, balsam, pea plants, chickpea, within 15 to 20 minutes by the utilizing of 10% sugar solution. 10% sugar solution, the pollen tube will be germinate, and all the interaction, all the reactions which is occur after the pollination until the reaches of in female embryo sac or female gametes, till the female gametes, these all the events are included in the pollen pistil interaction. So that is the formation of pollen tubes, transferring of gametes from pollen grain to the female gametophyte, that is the embryo sac. These are included in the pollen pistil interactions. Then artificial hybridization. Normally, self-pollination and cross-pollination is of natural. Sometimes the plant breeders transfer the pollen grain from one plant to another plants for the formation of different hybrid plants. That process is known as artificial hybridization. During the artificial hybridization, following events is there. One is of emasculation. Emasculation is the process that is the removal of immature anther from the bisexual plant. Removal of immature anther from bisexual plant. If the bisexual plant is there, flower is bisexual, the anther is removed before maturation. In this way, that prevent the self-pollination. If we remove that immature and then after begging is there, begging means suppose this is the flower, female parts, this one is of male parts. So before maturation these male parts is removed and then after it is covered with the bags. Why it is covered? For prevent the uh, transfer of undesired pollen grains. Undesired pollen grains will not reaches on this stigma. Whenever this stigma or female part is matured, uncovered that this flower and dusting of desired pollen grain is there. Dusting of desired pollen, we require particular type of hybrid plants. So particular types of pollen grain will be dusted on this stigma and then after fertilization is there. This is the artificial hybridization, means cross made between two different plants which is required by the particular breeders. So through the process of artificial hybridization, we can produce hybrid plants that having some special character, some good nutrient quality, some nutrient quality or other features like ornamental values or any fruit values or size of fruits, size of grains. If they are there, so they increase all these characters with the help of artificial fertilization. So after the dusting, again the bag will be or flower will be covered with the bags. So another pollen grain will not reaches on that flower. In this way, the plant breeders made the artificial hybridization and produce good quality of plants and good quality character, seeds, fruits, and other things. So today we discuss mainly that three things. One is of outbreeding devices, the devices which prevent the self pollination and increase the cross pollination. Another one is of pollen pistil interaction, the interaction between the pollen grain and pistil from transfer of pollen grain to before fertilization. These are the post pollination changes. And third one is of artificial hybridization. Through the artificial hybridization, the plant breeders improve the quality of fruits, quality of seeds, or quality of different plants. They produce desired characters in the plants.